So, yes, it's my pleasure to be here and to be part of this event on variation and variability because it's a very important topic, not only for the industry, but also for research. I would like to tell you a little bit about a relatively large collaborative research project called Grain Up that was recently finished. Which is a research project, or was a research project that focused on nutritional characteristics of cereal grains, funded by the German government and co-funded by the German feed industry, and where several research institutions were involved. I would like to give a specific focus on digestibility data here. And to start with a few statistics, just to remind you the amount of cereal grains that are produced. Of course, part of this is for human consumption, but quite a considerable part is for animal feeds. And I took a few values here from a German statistics that shows the total amount of cereal grains that were used in 1 year. And the estimated amount of crude protein and the estimated amount of phosphorus that is contained in this amount of cereal grains. And what you can see here are the corresponding values for soybean products and for rapeseed products. Of course, they were lower in regard to total amount, but look at the values here for the crude protein and for the phosphorus. There were lower or not higher, at least, than the amounts that are contained in the cereal grains. That means, of course, we consider them mainly as energy sources, but in terms of crude protein and phosphorus, they are at least as important to know about and to be characterized. And that is why this presentation will give some focus on amino acids and on phosphorus. So the objectives of that research project were to provide more actual data on the current qualities of cereal grains. We wanted to focus on the variation that exists between different grain species, but also within one grain species. And as I said before, one important focus was on determining digestibility values in different species. We will focus on pigs and poultry today, but ruminants and horses were also considered in that project. And linked to that were attempts to improve in vitro and other predicting tools, because we are facing the situation of animal studies becoming more and more difficult to conduct, at least in the EU. And in the meantime, more than 20 journal publications emerged from this project. And if any of you is interested in any details and getting copies of this PDF, I have them all with me. So if you want, please approach me in the coffee break or later, and with your USB stick, and I can give you all this information. So there were a total of, as you can see, 134 genotypes or cultivars, if you prefer that, of cereal grains with this distribution among maize, barley, wheat, triticale, rye, and oats. And there was a specific focus on the genotype-derived variation between these grains. So the selection was made by our collaborating partners from the breeding industry. And the important point is, and that is very important when we see the results later, to keep this in mind, that all the genotypes from one grain were grown under identical conditions on the same place, using the same fertilization and agronomy details and all these things on the experimental research station of our university. And you see the amount that was harvested per genotype, and that was largely different depending on what animal species these genotypes were used in. And what you can see here is just an impression of what has been done in the field. You can see different things. One thing is that Hohenheim University is located in a very nice surrounding. Yeah, you can see that all these things have to be done on a relatively small scale, experimental kind of scale here with the seeding of the grains, and then you can see the stripes here of different colors in different stages of vegetation. Then this harvesting procedure here also with these very small machines. So this has been a tremendous amount of work done by our colleagues from the research station for all these samples. So when we come to the results now, this is a very simple beginning, and it basically picks up what we have heard before, also basically confirms what is known for the proximate composition of the different cereal grains. Here are only the mean values for starch, fat, fiber, crude protein, and ash. And yeah, as we all know, the starch, of course, has the highest proportion here, and we do have some differences as expected also in other nutrients. I wanted to explain you, but I can skip that because we have heard that before, that especially in regard to starch, the question of method standardization, whether it is a polarimetric method or whether it's an enzymatic assay, is extremely important, especially when it comes to comparing data between different studies. So, as I said, all this information, all detailed information for each individual genotype is fully published. So we don't need to talk about too much. Now, and if you are interested in that, it's all available. So when we come to the crude protein fraction now, here again, in principle, we see confirmed what we know from all the textbook values and from other studies. Please note that these concentrations are given on dry matter basis here. The crude protein concentration in maize considerably lower than in the soft grains. But you can see on the first glance that there also is quite considerable variation within one grain, genotype-derived variation within one grain, although fertilization and everything was standardized. When we have a look at the oil or ether extract or crude fat fraction, we see the known higher values contained in maize and also in oats, lower than in the other grains. Again, some variation, and I would like to point you to this extremely high variation here for the maize, but that is simply because some so-called specialty maizes were contained in this data pool, and they were bred for high oil content for different other purposes, not only for feed purposes. When we now go a step forward and look to the amino acid profile of the grains, this is expressed here as grams per 100 grams of protein, the lysine concentration. Then we can see a clear differentiation between the different grains. We do have relatively low values here for maize and for wheat, and the highest for oats, considerably higher in the lysine content in oat protein, but again, substantial variation within one grain. And this picture is totally different when we take, as a next example, methionine, where we have only a differentiation between maize on one hand and the soft grains on the other hand. That means that we have, depending on which amino acid we look at, we have completely different relative values here among the grains that we consider. So the question is whether any correlation could be seen or relationship between the crude protein content of the seeds and the lysine content in the protein. When all data are taken together here, you can see the clear differentiation again between different grains. But there seems to be a trend of a relationship saying that roughly 0.1 grams of lysine are contained less, which each 10 grams of crude protein being higher in these genotypes. Please keep in mind, again, this is not related to differences in crude protein concentrations that are caused by different fertilization, for example. This is only caused by the different genotypes that were used in the data pool. And the picture is similar here in the other example of methionine, where the relationship is even a bit stronger between the methionine content in protein and crude protein content of the grain. So, interestingly, and this is from another study, this is not from this GrainUp project, but I would like to show you this study because it shows some conflicts of interest, so to say, between lysine content, increase of lysine concentration in the grain and digestibility. These are data for triticale genotypes, and here they were produced either under normal nitrogen fertilization regime or with additional extra nitrogen fertilization. And when we have a look here at the lysine concentration in the seeds, then you can see here a clear effect of nitrogen fertilization on lysine concentration in the seed. And these seeds were then tested in cystectomized laying hens. This is the animal model that we often use for amino acid digestibility studies in poultry. Then you can see that the digestibility of lysine in all three triticale genotypes, the digestibility of lysine was significantly lower. Meaning that we may get some more lysine in the seed through fertilization, but when we have a look at digestible content of lysine in the seeds, it was not different. So we always have to keep in mind that when we change one trait, we may have negative effects on another. So as I said before, we also had a closer look at phosphorus, and now we get back to the GrainUp data set. You can see the total phosphorus concentrations in the different grains. Again, well known that maize has a relatively low total phosphorus content, but we also have the differences here between the different soft grains, and again, variation. And here, because sometimes people ask how does this look against established table values, these are values from the NRC PIC booklet, where more or less the mean values are relatively close to what has been found in this data set, with the exception of barley. But I mean, it is as it always is when you compare data from single studies with table values. They may fit or they may not, depending on where this data come from. So we should perhaps not pay too much attention to it, and only always keep in mind that table values cannot tell us the whole story. More important these days is becoming the question of what proportion of phosphorus is contained in phytate form, and you can see here that an increase in total phosphorus concentration is associated with a certain increase of phytate phosphorus concentration, and this is something that is not investigated in much detail in the, or not considered in the tables that are around. You usually have only these mean values here, and you can see that this not at all reflects the variation that we have in phytate phosphorus concentration, and having in mind what big pressure we have on phosphorus excretion in some parts of Europe, then this is extremely important information for the fine-tuning of the diets of pigs and poultry. Okay, so let's now come to digestibility data. I'm watching my co-chair asking whether I still have enough time, and this will be on this will have a focus on digestibility of amino acids and on phosphorus. And I would like to start with a few explanations and on how we studied amino acid digestibility here for the poultry side of the story. We did that with laying hens, and these laying hens were all cystectomized. You can see how big the CK of laying hens can be, and that is a considerable room of fermentation producing bacterial protein that is no longer produced when these are removed. So 4 trials were conducted with these cystectomized laying hens, and each trial investigated 20 genotypes of the grains that are displayed there. And they were all conducted in the same way, using a basal diet that contained some maize starch, and this maize starch was replaced by one of the grains. And then linear regression approaches were applied to estimate amino acid digestibility of these genotypes. And in pigs, we also used a well-standardized approach using ileally cannulated growing pigs. You can see the surgically fitted cannula here. Again, 4 trials. In this case, only 8 genotypes of the grains, because it was not so easy to investigate so many genotypes, as many of you can imagine. And the so-called direct method was applied, where the cereal grains were used as the sole source of crude protein in the diet, and ileal digester was sampling according to a standard protocol that we use since many years in Germany. So when we start with the pig results now, you can see I've selected a few amino acids just to show the principle. What you can see here is the amino acid, the digestibility for lysine, methionine, threonine, and tryptophan for the 4 grains here. And please consider that the y-axis has different scales for the different amino acids. And there were very clear differences in digestibility of all the amino acids between the grains. And the rye was very low, particularly very low in digestibility. You can see lysine digestibility here. Lysine digestibility was on an overall relatively low value here for all grains. But the variation more or less was the same and in the same direction for the different amino acids among the different grains. And when this is compared to tabled values, and here again the pig data from the NRC were taken, then it is it becomes very clear that these values are considerably lower than these older tabled values, and the question was and still is what the reasons for this could be. And it probably has not so much to do with the changes in the grains per se over the years. It probably is related to the method and to the better standardization of the methods over time. I mean, these values that were tabled in 2012, they have been determined, I don't know, the 20 years before or even longer. And approaches and assays were not so well established at that time. And you can see the values here again for barley from the NRC and from this Grain Up project, but also from other studies. This is from Hans Stein's lab here for barley. And it was not only in the Grain Up dataset, but also in the US work here that digestibilities were lower than the older values. And the picture was very similar for wheat, where also this more recent data, irrespective of where these data were obtained, were lower than that what is contained in the older books. So there is something going on the assays. Now, after we have seen this variation, the question, of course, is what could be the reason for this variation, particularly within one grain. And as I said in the introduction, we were also interested in approaches to estimate or to predict this variation. And a lot of statistics has been run on this kind of data. And of course, the question was, is there any relationship between amino acid digestibility, in this case, as an example, lysine digestibility, and the NSP fraction. And this is the total NSP content here displayed on the axis here. And I mean, you can see for wheat and for barley and for triticale, there was no relationship, no significant relationship between amino acid digestibility and NSP content. The only significant relationship was detected for rye. So this does not mean that such relationship between NSP content and amino acid digestibility does not exist. It simply means that we could not detect it in most of the grains here, and that is most likely related to the relatively low variation in NSP content that we here had within one grain. And that again brings us back to the standardized environmental and agronomic conditions that were applied here in this experiment. So when we also have a look at the results of the laying hens now, it's pretty similar, but on a higher level. Especially for lysine. Values here for wheat, rye, triticale, and maize. Barley was not investigated in laying hens. Again, the rye with discouragingly low digestibility values here, but overall relatively high values for the digestibility of the amino acids in the laying hens. And that is not so easy to compare with other data from literature because, well, there are hardly data available. And if they are available, they usually are based on different methodological approaches. And we all know that details of amino acid digestibility highly affect the outcome of the experiments. But I think we can say that this study or these results in principle confirm that the level of amino acid digestibility is relatively high in cereal grains. And what I can do is making a comparison with this cereal grain data with some other data that we have generated using the same experimental approach. So this the cystectomized laying hens, and when we look at the mean values here for lysine digestibility, this is what has been contained in the graphs already before. We see this around 80% for wheat and maize, slightly lower in triticale and much lower in rye. And when we have a look at rapeseed meal, it's also not very high, but values are much higher, for example, here in lupins and peas that were investigated in this animal model, and also for methionine, the data are shown here. Methionine is of relatively high digestibility in cereal grains, and even higher than the digestibility here in the legumes. So again, for this data, we tried to play around and run a few statistics on relationships between digestibility and certain specific fractions analyzed in the grain, and tried multiple regression analysis in an attempt to predict amino acid digestibility. And for this, all the data that were chemically analyzed were grouped in these 8 pools here, and then the computer played around with these data. And what I can show you here is that using methionine again as an example, when you select here these different number of variables in these multiple regressions, then a relatively high accuracy of prediction can be obtained for the estimate of methionine digestibility. But have a look at these fractions that are considered here. This is something that cannot be done under routine conditions, and that means that this is, at least at this stage, an artificial situation and cannot be applied for predicting amino acid digestibility in laying hens. So combining several variables that were analyzed may lead to an acceptable accuracy of prediction, but the selected variables are neither useful to explain this variation, nor are they of practical relevance because too difficult to determine routinely. Another interesting question was, how does it look like when we compare the pig and the laying hen data? This again only lysine as an example. The laying hen data here and the pig data here. We can do this comparison because these digestibility tests were done with the same batches of feed. And what you can see here is that yes, if you take all the data together, there seems to be quite a fair relationship. But when you look into each individual grain here, then these relationships are not existing. So you can see the R-square values here for wheat, triticale, and rye. Only when all data are considered together, relationship can be predicted. But again, we have to consider here that the range that we have had in this data set was relatively low. And last point here, as I said, we also tried to develop or improve in vitro assays. So on all the samples, the Boysen and Fernandez-based in vitro nitrogen solubility was also applied and measured here. And this is for the pig data, and this is for the laying hen data. The relationship between this in vitro nitrogen solubility and the determined digestibility. In that case, again for lysine. And there was no significant relationship with the exception of the rye data here for all the other grains. And you can also see here for the laying hen data that this is so far not a really satisfying relationship. Still more work to do on that. So a few words finally about digestibility of phosphorus, again for pigs and for, in this case, broilers. For the pig, we have since many years a standardized assay to determine digestibility of phosphorus on total tract basis of feed raw materials. And for broilers, we have the WPSA protocol to study pre-cecal digestibility. And when we immediately dive into the data for the pigs, this has not yet been published, done by the colleagues at Bonn University, wheat, barley, rye, and triticale. Again, 8 samples investigated for phosphorus digestibility, and you can see the mean digestibilities here, 60% for wheat with this variation among the 8 samples. Barley in the known range, rye surprisingly low, taking into consideration the high phytase concentration of this grain, and triticale somewhere between wheat and rye. So again, we see also for phosphorus digestibility this high variation here, and this could not be explained by phytase activity alone, and it could not be explained by phytase activity and phytate proportion together. And for the broiler data, yes, thank you. We have the pre-cecal phosphorus digestibility here for 8 samples of triticale and for 8 samples of wheat. If we look at the triticale samples here, the orange ones, you can see that they are somewhere around, what is it, 80% for some of the samples, and others were down to 60. And the wheat digestibility was lower. It was around 40% for 4 samples, and between around 60 and 70% for some other samples. And I choose this way of expression depending on plant intrinsic phytase activity to show you that, well, there was quite considerable variation in the intrinsic phytase activity of the seed. But this intrinsic phytase activity of the seed could not at all be used to explain the differences in phosphorus digestibility found here. So there are other factors relevant, and intrinsic phytase activity in broilers is not so important. So just towards the end, only to mention, if there is anybody in the audience who should be interested in ruminant research, ruminants were also considered in this project, so there are data available on ruminal degradation of protein and starch, and you can get this data if you want. So to conclude, the results from these animal studies allow for a more precise consideration of protein and phosphorus values in diets. We had seen in the previous presentation that there is a lot of variation in chemical composition of all the feeds that we use, including grains, and we have to consider that we also have this variation on the digestibility side. So running chemical analysis is not solving the problem alone. We have seen in the pigs that amino acid digestibility differed largely between the grain species. Overall, the values were lower than reported in feedstuff tables, but are in agreement with more recent publications. This points towards assay relevance here. In hens and in broilers, we had again and also differences in amino acid digestibility between the grain species and among the genotypes. The amino acid digestibility is relatively high in laying hens, except rye, and phosphorus digestibility also is relatively high. And so far, and this may be the little bit, well, perhaps, I don't know, frustrating or disappointing message, that so far we are unable to explain this variation of digestibility data simply by chemical characteristics of these grains, but that is something we can continue to work on. With this, I would like to thank you for your attention and look forward to your questions.